So, good afternoon, everyone. Welcome back to part 2 of Professor Scott Linderman's seminars at Ideas and ARC. And today he's going to talk about state-space models, more recent research. So without further delay, Scott.
All right. Thank you. And thanks everybody for coming back for day 2. Yesterday was more about, let's make sure this is still working here. Nope. Try turning it off and turning it back on. There we go. Yesterday was more about the foundations of probabilistic state space models. So we covered some motivating examples and some of the sort of textbook probabilistic state space models, the type of stuff that you can find in, I really like Kevin Murphy's textbook on probabilistic machine learning. And you'll find many chapters on HMMs and linear Gaussian dynamical systems, nonlinear models, switching linear models, all that type of stuff, as well as many of the algorithms that we talked about yesterday, or at least we started to talk about yesterday for performing parameter estimation, i.e. learning, and inference, i.e. state estimation in these types of models. So we briefly touched on EM, expectation maximization algorithm, some of the core message passing algorithms. Is there another mic on? I don't think that's from me.
Sorry, I think it was me.
It's you? And, so we talked about message passing algorithms for state space models, for like chain structured graphical models. And we saw how this simple recursion, the forward message passing algorithms applied to discrete state HMMs, to linear Gaussian models. But for anything more complicated than that, we had to start making some approximations. So we talked a little bit about some of the ways you can make those approximations using extended or unscented Kalman filters for nonlinear systems. And then more generally, using techniques like sequential Monte Carlo or variational inference. Today, we're going to dive into a bit more on both the modeling front and the algorithmic front. So I had an ambitious schedule for all the things. I would love to tell you about all the work we're doing in my group, but I think it's better to maybe try to focus today's talk on 3 relatively short stories. 2 of them are about better models for state space or for sequential data. One of them is about better inference algorithms. So we're going to do this in a somewhat nonlinear fashion, in keeping with the nonlinear dynamical systems that we've been talking about. We'll start with some of the recent work we've done on time-warped MoSeq for segmenting behavioral data. We'll then switch and talk a bit about algorithms for sequential Monte Carlo using smoothing distributions as targets, which we call SIXO, and then end with some very recent work on what we call state space layers, which is an exciting, relatively new class of models that are proving to be really useful for sequence to sequence mappings, both in neuroscience as we found, but also much more broadly in machine learning applications. So just like yesterday, it works best if you have questions, just raise your hand, shout them out. I'm excited to field those questions and to try to answer them to the best of that I can. So don't be shy about that. Okay, cool. So without further ado, let's dive in and let's start by talking about what we call time-warped MoSeq. And just to jog your memory from yesterday, we started with this application of trying to segment the natural behavior of mice in a freely moving in an open field in this bucket here, as we see. This is a depth video. For those who weren't here yesterday, this is a Microsoft Kinect looking down on the mouse. When it rears up on its hind legs, it gets closer to the depth sensor, pixels get brighter. And so what we'd like to do is to take this video, first of all, find the mouse in each frame of video, rotate it so it's pointing to the right, and then see if we can try to segment this video into stereotyped, simple, reusable behaviors. The reason we think that's a decent model as a starting point is that if you just do some random projections of this video to look at this as a high dimensional time series, like what you see on top, you can see pretty clear change point structure just emerging from these high dimensional projections. We see that as well in the lower dimensional representation, i.e. the embeddings into the top 10 principal components on the bottom. Now, how does MoSeq work? How are we thinking about this model? Well, instead of looking at that as a time series of principal components, imagine that we just looked at a scatter plot of all the places in PCA that are visited during this roughly hour-long video. So each dot in the span of the top two principal components corresponds to the embedding of one video frame. So some of these correspond to the animal on all fours, others correspond to the animal rearing up on its hind legs. And these aren't just any 4 dots. It turns out this is a sequence of video frames that unfolds over the span of about a couple hundred milliseconds. Now, one question that you might ask is this little snippet of trajectory through this 2D space a once-in-a-lifetime event? Is this the only time the animal ever performs this particular sequence of movements? Or is this something that's stereotyped, something that happens again and again and again that we might expect to find repeated instances of in this data? And indeed, if we look throughout this recording, we can find many instances of little trajectories that look very similar to this one, tracing out paths through this PCA space. This makes us think that maybe this behavioral video can be thought of as a bunch of stereotype trajectories through PCA space. Now, is it unique to that particular trajectory? No. We can find many instances for basically any trajectory that we see, and we can find many instances of it. There's very few like once in a lifetime occurrences, or at least once in an hour long recording occurrences here, it seems. So the idea, the first critical idea to this MOSEQ model is that maybe we can approximate each one of those short trajectories as a realization of a simple dynamical system operating in this PCA space, namely a simple linear dynamical system. For each one of these trajectories, we could perhaps approximate it as a realization of a linear dynamical system of the form here. Xt plus 1 is ax plus b. Different a's and b's correspond to different vector fields like we talked about yesterday. So we expect that we're going to need a lot of these in order to model an entire trajectory, but at least each short trajectory, we think we can model as a simple linear system. So the second key idea is if we can model a short trajectory as a linear system, maybe we can model an entire video, i.e. a long, many hundred or many thousands of frames of video as a sequence of short trajectories. And that's what we call these behavioral states here. So a transition from red to blue to yellow corresponds to a particular path through this PCA space, maybe walking, pausing, and then rearing up on the hind legs. To formalize that, like we talked about yesterday, let's try to write this out in terms of a graphical model. So a graphical model, again, that has nodes. These correspond to variables. The filled in nodes are ones that we observe. Unfilled nodes are latent, ones that we wish to infer. And it also has arrows that tell us about the conditional distributions of one variable given its parents. So here, in order to simulate the next position in PCA space, you need to know where you started and what state you're in. Moreover, you need to know the parameters associated with each of those discrete states, because those are the a's and b's that tell you about this vector field. If you knew these 3 things, you could model a trajectory through PCA space. But the other key observation here is that the sequence of discrete states, behavioral states, isn't just random at each time frame. There's some temporal dynamics to these discrete states as well. If you're in the walking state for frame 1, it's very likely you're going to be continuing to walk at frame 2, so on and so forth. And we capture that with this transition matrix as written up here. And then finally, the way that MOSEQ, at least as originally implemented, had this fancy non-parametric prior, because remember, one of the questions yesterday was, how do you know how many discrete states you need? Well, there's a number of ways to try to answer that question. No way is necessarily perfect, but one way to try to answer this question is to say, I'm going to try to be agnostic about this. I'm going to build my model with the capacity to infer the number of discrete states that's best matched to this data. And one way to do that is with a Bayesian non-parametric model, like a sticky hierarchical Dirichlet process. Now, if you put all these things together, this is what in the machine learning community and the statistics community we would call a sticky hierarchical Dirichlet process autoregressive hidden Markov model, or a SHDPARHMM for short. So Emily Fox, who is actually, she just recently joined the stats department at Stanford. She's amazing. Did a lot of work on this when she was a grad student and postdoc. As you can imagine, this name did not necessarily catch on amongst neuroscientists, and instead they simply refer to this model as MOSEQ, motion sequencing. Now, while they're at it, behavioral states is a little blasé. let's give this a more evocative name. We'll call these behavioral syllables. By sort of analogy to the building blocks of natural language, we think of these short stereotyped behaviors as the building blocks for complex compositional sequences of behavior. So what do you get out of MoSeq? Well, we showed a couple examples yesterday. When you segment this video, we can go in and we can look at one instance of, say, the blue syllable, and we can see what the mouse was doing in that instance. When the dot comes on, it indicates the start of that syllable. And here we might say, this is a darting related behavior. The dot comes on, the mouse pokes forward. Now, again, is that a stereotyped thing, or is this, representative of a repeated action? On the right, we'll show a video where we've highlighted 50 instances of what the model identifies as the same little behavioral snippet. And you'll see the dot comes on, all the animals dart forward. They persist for different amounts of time. These syllables aren't defined as like a particular duration long. They're defined by dynamics that can be employed for sort of an arbitrary number of time steps. We're looking for syllables that unfold on the orders of couple hundred milliseconds at a time. That's about what a linear system seems capable of capturing. Yeah.
So this model is operating on the sector version of the mouse image, right? So it's not using any of that like spatial information where the mouse is actually like translating across the bucket. Is that what's happening? So it's like picking up on the structure without actually knowing the mouse.
That's true. So the question for the Zoom audience was, the model is just looking at the centered and rotated videos. It doesn't know anything about the absolute position, heading, et cetera, of the mouse. That's absolutely correct. So the ways that it finds these, these syllables that correspond to forward movement aren't because we've given it absolute position and velocity. It's because when the mouse darts forward, it engages a certain particular pose and a sequence of poses that are employed during walking. And so we don't find syllables that are, we rarely find, I don't want to say absolutely, we never find syllables that have both still and movement, but I think it's very uncommon. Usually you find syllables that are, similar movement speeds like we see here. All right, so this is a darting behavior. Sorry, my clicker's having a little bit of trouble sometimes. That's only one of many different types of syllables we pull out. Often these models will find around 50 to 60 syllables to capture about 95% of an hour-long video. So darting, running forward, grooming, scrunching, rearing, trying to escape the bucket. I showed you a couple of these videos yesterday as well. One of the reasons why we're interested in segmenting natural behavior like this is because, we're interested not just in behavior for behavior's sake, though that is, certainly a valid biological question. Ethologists who study natural behavior and the evolution of behavior are really interested in those questions. But as neurobiologists, we're interested in the underlying neural circuitry that produces these natural movements. In particular, we're interested in this circuit that involves cortical signals extending down into the striatum, which is part of the basal ganglia, and ultimately en route to descending pathways through the brainstem and out to your muscles to move. So this has been a long-standing collaboration with the data in the Sabatini labs at Harvard Med School. Now, what they are able to do, and by the way, Jeff's not here today. Jeff Markowitz, who is newish faculty here in BME, is really the one whose work I'm showing here. He did these really amazing recordings where he put a microendoscope into the striatum, the dorsal and lateral region of the striatum of this mouse, and let it run freely around in the bucket. And then did the same type of thing, depth video recordings as the mouse ran around. But now, in addition to getting behavioral syllables, we also get these time series of calcium transients, which are indicating underlying spiking activity of these neurons. You can see that in this video here, where these neurons are fluorescing as the mouse runs around. And we can get this time course of behavioral states that are estimated, as well as neural activity that's going on at the same time. There's a small technical hurdle that we had to clear here, which is that a lot of our analyses were based on principal components of these video frames like we see up top. But now these frames are dominated by this cable that's swinging around overhead. So if you just do PCA on that, like the top principal components don't correspond to the mouse, they correspond to where the cable is. That's the largest variance contributor to these videos. So what we did is we were able to figure out where the cable is, because it's the tallest thing in the field and in the frame, and then basically mask that out and then use basically dimensionality reduction with missing data, factor analysis with missing data to try to find that low D embedding to run this MoSeq model on. When we look at the neural time series that are happening around the onsets of different behavioral states, we find these beautiful sequences of activity in both the direct pathway and the indirect pathway through the striatum. And we find different sequences of activity for every different syllable that we find. So in each panel here, the neurons are permuted to best show the sequence of activations across this population, but the permutations are different depending on whether it's a rear or a scrunch, et cetera. And we find that, somewhat contrary to the theories that had been kicking around before this, that it seems like both of these pathways are in a very complementary way encoding the onsets of these different types of stereotype movement. Again, if we then order these neurons based on the pattern, the permutation that shows the biggest activation for scrunching, we see that when you look at darts or scrunches to the left, even very similar syllables like scrunches to the left, the pattern is very different. Likewise, when we permute by darts, the pattern of activation is not nearly as clear. It's really suggesting a somewhat syllable-specific coding of activity of each one of these individual behaviors. That suggests that maybe, given this neural activity, we should be able to predict on a moment-by-moment basis what the mouse is doing. And the way that we tried to quantify that is we said, all right, the model gives us 50 to 60 syllables. Let's start by splitting them in this, just using a very simple hierarchical clustering into two groups, which at the highest level ends up being sort of still movement, or still versus movement. And when you bisect at that level, you can predict what the mouse is doing based on neural activity with close to 90% accuracy. And we can do the best when we include both populations, not just the direct or the indirect pathway. As we go down this tree and we ask the model to make finer predictions, finer grain predictions about what behavior is being executed, prediction performance goes down, unsurprisingly, but it's still considerably above chance level of just random guessing based on the relative frequencies of these syllables. Now, this I think is an encouraging result, but it's also one that kind of led me to think a bit about some of the limitations of this approach. And in particular, as we get down to the leaves of this tree, are these really meaningful distinctions of behavior? If the model, sure, can predict above chance, but is it really like, are these distinctions between syllable 1 and syllable 2, meaningful from this bigger picture perspective? And one reason why they might not be is if we're effectively fitting a discrete state model to data that lies on a bit more of a continuum, and just segmenting because the model's got limited capacity for any individual state. So given enough data, it's just going to end up, drawing these discrete boundaries in places where we more naturally would have defined these to be the same syllable. And one reason to maybe think that's happening is if we look at a handful of just 6 syllables here, it's really hard for me, even after many years of watching videos like this, to tell you what's different about these 6 syllables. The model says these are definitely different. If you give me fewer syllables, I'm going to pay for it in terms of lower log likelihood of the data or held out data. But I can't give you a human interpretable name. Maybe this one's slightly slower. And that was a little bit faster. This one persists a little bit longer. But to me, these all look like darting behavior. Maybe if Jeff were here, he could tell me there's a real fine-grained distinction. But I think he'd be hard-pressed to say what it is that the model is queuing on to distinguish these syllables from one another. And one idea that we've had, oh yeah, sure.
So if you look at the corresponding neural activation for these different syllables, is that similar or is there distinction there as well?
So I think I don't have the complete story for you. I think that as the syllables get more similar to one another, the neural activation patterns get more similar. However, I showed you that one scrunch versus scrunching to the left. Like these differences in orientation do have, at least at that level, we can see big differences in activation. But I don't know for these 6 individual syllables here if the neural activations are how similar or different they are. I think they're probably fairly similar too. We'll come back to that in a little bit because one of the things that we thought might be going on here is that there really is a continuum of syllables and that the model is just discretizing them because that's all that a HMM can do. And one thing that we thought might be contributing to that continuum is that maybe there's some extra continuous latent variable that needs to be added to this model in order to give these states a bit of extra capacity. So what we did, and this is work by Julia and Leah in my group that we presented at NeurIPS last year, is we extended this MoSeq model with this extra, just one-dimensional continuous latent variable that we call tau. Now, the question is, how do you link tau and Z to the dynamics through PCA space? There's not, this graphical model doesn't say anything about what the functional form of that relationship is. That's a choice, a design choice that we have to make. And our thought was, well, maybe let's let tau govern the time constant, the speed at which these syllables unfold. And a simple way to do that is to say that the next position in PCA space, the next observation xt, is wherever you were previously, plus this update, a times the difference between the previous state and some mean, and then scaled up or down by tau. Now, this is effectively the same model as we had before. It's just written in a different way such that it emphasizes how this time constant can speed up or slow down the delta, the update from your previous position in PCA space. So when tau is 1, you may follow these dynamics, but when tau is less than 1, you slow down, you don't go as far. Tau is greater than 1, you traverse further, but along the same path. So it's effectively like how many time steps have elapsed between one frame and the next, or how much effective time has elapsed. And that's why we call this model time-warped MoSeq, because it gives this extra capacity to continuously modulate the speed of syllables. So when we run time-warped MoSeq again on the same data, we find that it lumps a lot of those darting-related behaviors together into one discrete state, and then it distinguishes them instead on the basis of their speed. So on the left, we've got a very slow syllable. On the right, we've got a very fast version of the same syllable. So let me see if I can play these one at a time. Right, so this was a slow dart. This one's getting a little bit faster, and then hopefully fastest. So we think that that gives us, at least at a high level, the first pass sanity check. It looks like it's doing about the right thing. We can do the same for syllables that don't have an overt speed measurement associated with them. Things like grooming related behaviors, turns out, can also be performed more or less vigorously. And the model also picks up on that as it attributes it to this latent time constant that we've given it. And a more quantitative measure of what this model is doing is that if we look and we see how many syllables do we need, how many discrete states do we need to achieve some target level of held out log likelihood, we can obtain the same performance with almost half as many syllables using this time warped version. So it's scaling back the number of discrete states by a factor of 2. And I won't show it here, but it also facilitates comparisons between mice from different groups. Maybe I will show this a little bit, let's see. But for example, if you give mice amphetamine, then in general, they speed up. But do they speed up all syllables equally, or do they just speed up particular running related syllables? It looks like it's a sort of uniform shift in the speed or the vigor at which syllables unfold. And that's regardless of whether it's grooming related behavior or walking related behavior. This is exactly what I just showed you. Where, except for maybe one syllable, that's just pausing related behavior, we see this sort of upregulation of speed across all of the syllables, even though their usage is exactly the same. So if we zoom in on just rearing related behavior, under amphetamine, there's this nice and very significant shift in the right of this distribution. Same is true for darts. And this is what I was saying about it. It's happening over all syllables. There's this uniform shift where, except for one of these syllables down here, which was more or less pausing related, the average time constant that we infer is higher across the board for these amphetamine groups, which is kind of a cool finding, and also I think a nice further confirmation that, yeah, this time warped MoSeq is doing something very sensible in this setting here. Now, why is speed particularly interesting for us? Well, if we go back to the neural circuits that we were studying, speed and vigor have been postulated as one of these really important roles for dopamine, a key neuromodulator that's impacting these circuits in the dorsolateral striatum. Many of you have probably heard about dopamine in the context of reinforcement learning and carrying and conveying reward prediction errors. Turns out that dopamine projecting from SNC to the dorsolateral striatum has a different role, or at least it plays another role that's more related to movement. These are the cells in SNC that are dying in Parkinson's disease and leading to the inability to initiate movements and things like that. So we're really interested in how dopamine that's impinging on these circuits in the dorsolateral striatum is modulating the speed of movement and changing, perhaps which types of movement the animal executes. And so what we've done, again, very recently with work that, again, was led by Jeff in collaboration with Wynn and Maya in the Data and Sabatini labs, is to go in and record not calcium transients from individual neurons, but rather the bulk activity of a dopamine fluorophore D-light that's been expressed in these same neurons in the dorsolateral striatum. So here what we get out is, again, it's similar. Here's the mouse running around. This time the cable is a lot smaller because this is just a fiber-photometry recording, but we get this one-dimensional trace that's correlated with how much dopamine is floating around in the vicinity of these neurons that are in the dorsolateral striatum alongside the syllables that are being executed. And so we're looking at this D-light signal through the lens of time-warped MoSeq, and we're asking, to what extent is this D-light signal correlated with the inferred time constant and the inferred syllables, and maybe even the principal components of this mouse body posture over time? And this is still very early stages of this work, but already we're starting to see some pretty interesting relationships where, at least on very short time scales, this dopamine signal that's being measured seems to be anti-correlated with this inferred vigor signal, which is somewhat contrary to what you might expect. If you look over long time scales, you see positive correlations between dopamine and vigor of movement. On short time scales, that seems to be inverted. And we can see that in particular when we look at transitions between speeds of movement. So I think there's a lot of interesting work left to be done here. This is the type of stuff that we're doing right now. But it's already pointing to how these models that we're building out to capture and dissect behavioral data in a more refined way, not just discrete syllables, but continuous degrees of variation between behaviors, are allowing us to go back and look at these neural signals of interest through a new light. And I'll just end this little section of the talk with one shout out to very recent work that's also extending this MoSeq model in a somewhat similar way. So MoSeq is this simple model that we started with, and in this first part on time-warped MoSeq, we added another layer of continuous latent variables. Here, we were interested in how we could apply the same type of model, not to depth video, but to key point data. And in order to do that well, we added a bit more of a noise model, a bit better noise model to this, where we allowed for mappings from these, rather than just doing PCA on the data to start, we treated these latent continuous embeddings as latent variables to be inferred. And when we fit this to key point data, we can find really nice syllables that correspond to different types of movement, just as we had before. But the barrier to entry to using these techniques is a lot lower because you don't need to set up a Microsoft Kinect. I don't even know if you can still buy a Microsoft Kinect. That's maybe one of the first challenges. All you need here is a regular RGB camera. It doesn't even have to be super high frame rate or anything. Just the off-the-shelf cameras that you need to do pose tracking, which are widely used throughout neuroscience these days. So we're excited about this, and we just dropped it on bioRxiv a week or two ago. So check it out. It's called Keypoint MoSeq.
Yeah, it's all in LastPass.
Okay, so like for food.
Any questions on that? Yeah.
There's like the range of speeds that can be modeled for a particular.
Did Chapin share the card with you?
I haven't set up like
Like very fast.
Gotcha.
Like
Yeah,
very
you
fast
should
and very slow
ask
speeds,
him.
but the
You
results
just set
are
it up and then ask him to share anything.
Yeah, so the question, what's the range of speeds that you can capture with an individual discrete syllable? So here speed is defined relative to the sort of baseline speed. Tau equals 1 is our sort of reference speed, and then you can speed that up or slow it down by a multiplicative gain factor. We typically set that from like 1/3 x to 3x the speed. Usually once you get down to 1/3 x, all the syllables start to look like pausing. So the model starts to become kind of unidentifiable in that regime. But that's not like a hard limit of the model. Like it's just a practical guidance that we use when trying to set that limit. In terms of absolute differences in speed, I don't know the numbers off the top of my head for like how many centimeters a second or whatever the velocities of movement are here.
Then I guess like what are the transitions in the model between the taus?
So the only constraint we place on the taus is that they change, relatively smoothly. So you can't just go instantaneously from 3x speed to 1/3 x speed. You have to move through that at some with some smoothness. You probably noticed in one of the plots that the taus looked pretty stepwise. The reason for that is that since tau is just a 1D variable, it was simplest way to do inference in this model is to discretize tau on a fine grid from that limit of 1/3 to 3, and then just treat this as a big discrete model with a whole bunch of both discrete syllables and discretized speed levels. You could do this with continuous models. It just becomes like with a truly continuous state and then a posterior that is also continuous. It just is kind of annoying and not that much more insightful, we think. Yeah.
So you mentioned how this the presence of dopamine, there is this inversion between shorter time scales. So what does it mean?
Yeah, that's a great question. We're still, if you read this paper from Jeff that came out earlier this year, we talk a lot about what might be going on in this, the short time scales, like, is dopamine kind of flickering on the onset of a syllable and then off in the middle and then back on at the end, and the starts of syllables, maybe a little bit slower than the middle point of a syllable. There's a lot of things that are still left to be fully understood about this, and that paper has many more results on what we are speculations and some preliminary evidence about what we think is going on there. It seems like dopamine concentration and fluctuations are also correlated with the entropy of future syllables, or syllable transitions, I should say. So when you're sort of at a lower dopamine tone, maybe you're performing more stereotyped transitions from behaviors, and then when you start to increase that concentration of dopamine in these circuits, the transition rules seem to be a little bit more random. But these are all things that I can't do justice to here. But I would encourage you to take a look at that manuscript, because there's many cool things in there. I think there was one question on Zoom here. Let me see if I can open that. Did you observe any variation between mouse to mouse, or is all on 1 hour snippet of movement? Thank you. Yes, so we fit the model to data collected across many mice that are all age and sex matched and weight matched in order to make them so stereotyped as possible. And so all of the analyses that I'm showing you are pooled over many mice, though there is an interesting question about mouse to mouse variability. It's not one that we focused on in this experiment. Okay, so maybe there's going to be a bit of a discontinuity here as well. There's like continuous changes within section, discrete changes between sections. This one is a rather discrete jump where we're going to talk now more about algorithmic innovations. So the first part was all about innovating on the model side. Here we're thinking more about how we can do better inference and parameter learning in state space models, which is obviously a key part of this whole process. So this is what we call 6.0. And I want to highlight the work was done by Dietrich, who's a grad student, and Dietrich and Alan, who are grad students, Andy, who's a postdoc in my lab. And we call it 6.0 because it's smoothing inference with twisted objectives. And you got to kind of, well, there's a whole history here. There was a method called 5.0 before. 6.0 is the natural successor to 5.0. So the twist is the X there. And it's all going to boil down to the differences in the target distributions that we use when performing SMC. And if you don't know what that means, don't worry, I'm going to explain all of it. But at the end of the day, the takeaway is that this is going to be much more effective at producing posterior distributions by leveraging information that's yet to come in the future. All right. So as a motivating example, the application that got me particularly excited about this was this this application to voltage imaging data. This videos are created by Daniel Hochbaum when he was a grad student in Adam Cohen's lab back in 2014 timeframe. And so they're optogenetically stimulating these neurons using blue light at these dendrites here. And then, as they optogenically stimulate, it's driving current into the soma, initiating a spike, which when you see these videos, you can watch propagate throughout the cell. And it's absolutely beautiful videos. These are the types of things that you couldn't do with just electrophysiological recordings, because you can only get an electrode into maybe one compartment of the cell, usually the cell body. It's really difficult to make multiple recordings from one cell at multiple parts of the cell. But optically, if we can record light from many compartments all at once, we can start to see these propagation of action potentials like you see here. The trick is that the data doesn't actually look like this. This is this superposition of many spikes, thousands of spikes that are happening at slightly different delays relative to the sampling, the frame rate of this camera, such that they can go back and do this effectively like super resolution video. Very, very fine time resolution to look at what an average spike looks like. But if we want to do this, using raw data and infer on a moment-to-moment basis what the voltage is, we need some tools that can, do inference about voltage dynamics given very, relatively slow and noisy light measurements. And the way that we're proposing to do that is by leveraging all of our detailed biophysical knowledge about how voltage propagates through cells, namely the Hodgkin-Huxley model, and how that extends to multi-compartment models of neurons, where when you inject current into one compartment, it bleeds over, voltage propagates over to these other compartments, and sometimes initiates spikes in these downstream compartments by modulating which ion channels are open or closed at any moment in time in these compartments. So we've got these generative models for spike trains. The question is, if we only observed noisy, slow voltage recordings from these compartments, could we infer this underlying voltage trace and the states of all these ion channels that are producing these dynamics of voltage? In order to do that, we need to do inference, and we also need to learn the parameters of these models, because each compartment is going to have different compositions of ion channels. It's going to have different conductances between compartments. There's tons and tons of parameters that go into a Hodgkin-Huxley model. And also the dynamics of these latent states, the activations of different ion channels, the voltage at each point in time, these are all highly nonlinear in the Hodgkin-Huxley model. And then finally, the mapping that we have from states to observations, in this case, we'll typically assume a linear readout of the underlying voltage just with some additive Gaussian noise. But in general, you could imagine nonlinear measurement models. And so we'd like to have tools that work not just for Hodgkin-Huxley, but generically to perform inference and model learning in nonlinear dynamical systems like what I've shown here. And remember, these models really correspond to a joint distribution over the latent variables X, the observations Y, and just specify how that factors into a product of conditional distributions. Now, one way that you could imagine trying to do posterior inference, we didn't talk about this one yesterday, but this is a basic Monte Carlo method called importance sampling that you could use to try to estimate the posterior distribution over latent states X given observations Y. Now, just imagine we're looking at a one-dimensional latent variable X. Forget about the whole time series, just zoom in on a single dimensional latent variable and assume that the true posterior looks something like this blue curve. The idea in importance sampling and in the methods that'll build on this is that we're going to approximate this with a weighted collection of particles. So each particle is an atom located at some position. These are the vertical blue lines. The weights are the heights of these. So we're going to have a weighted collection that we want to look, in some sense, like this blue curve. And so the way that importance sampling works is that first we're going to draw locations of these particles from some proposal distribution. This proposal distribution we'll call Q. And in general, it could depend on the data that we're seeing. There's very few constraints. The only real constraint is that Q's got to be positive wherever the true posterior distribution is positive. Second, we're going to compute weights under this model. And these weights are going to be the ratio of the joint distribution, the thing that we specified on the previous page from that graphical model, divided by the proposal probability, this density Q. And then once we've got all the weights, we're going to normalize them by dividing by the sum. So first thing to note, all these weights are positive because both P and Q are positive, and Q is greater than 0 anywhere P is greater than 0. That's this absolute continuity requirement that we have up here. So these weights are always positive heights of these blue spikes. And then once we've done that, why have we gone to this? It's because once we've done this, collected these weighted particles, we can use them to compute or approximate expectations under the posterior distribution. And if you recall from yesterday, we argued that a lot of what we're trying to do in Bayesian inference boils down to approximating expectations under posterior distributions. That's exactly what you can do with methods like these. You just take a weighted sum of particles, and if you want to compute the expectation of f of x, well, it's just a weighted sum of f of xi at the locations of your particles. Any questions on importance sampling? Yeah.
Parameters of the P distribution?
The P distribution, at this point, let's just assume that we know P, but that's going to be an outer loop is going to be inferring the parameters of P. Likewise with Q, Q could also be a parameterized distribution that we could also try to optimize. In general, what would the optimal Q be?
P.
Optimal Q would be P of X given Y. Q is a density on X, right? But ideally, we'd like to propose from the distribution that we want to sample from. The starting point for this, though, is that the posterior distribution itself is not something that we can directly sample, because if we could, we would just do that directly, and all of our weights would be 1, and it would just be a Monte Carlo. It would reduce to ordinary Monte Carlo. So we're operating in a setting where we can't sample the posterior directly. So instead, we sample this thing Q that we've constructed and we've assumed to be easy to sample. And we're using that to get an estimate of this expectation with respect to P that we really want. Now, there's a lot more to be said about Monte Carlo methods and importance sampling. One thing to note is that when we have to self-normalize, these expectations are biased because we don't know the true normalizing constant for this joint distribution. And you can think of this normalization as effectively estimating that normalization constant, but they're going to be consistent in the sense that if we drew infinitely many samples, this expectation would be unbiased in that asymptotic limit. That's just a technical detail. Now, the problem with importance sampling is that the variance of these estimates, right? I said you can get you can get a consistent estimate of the expectation of f of x, but I didn't tell you anything about the variance of that estimate. And when designing Monte Carlo estimators, it all comes down to bias and variance. So here, the variance is going to become problematic. And you can think of this as like, if I'm proposing from a distribution over an entire sequence of latent states, this is a humongous dimensional space. And the probability that I propose a trajectory that really truly has high probability under the posterior is in general going to be quite low. I'm going to have Q is going to have to be really lucky to propose a trajectory that's going to have some reasonable probability under that posterior. And in general, you can think of that as saying, well, maybe at the first time step, a lot of my proposals were pretty good, highlighted in green. But as I go forward, I really need like an and of every time step. I need first time step to be good, and second, and third. And if each time step there's kind of a failure probability, by the time you get to the end, it's very likely that at least at some time in this chain, you're going to have made a bad proposal, things are going to break down, and the trajectory is not going to be really valid. And so that's what we see up here. We end up with maybe one good particle if we're lucky, and then that whole approximation of the expectation is based on just a single particle. It's not capturing any of the posterior distribution really, and so that ends up being super high variance. Now, what's the solution? Well, there's a method called sequential Monte Carlo that's built on importance sampling, but basically has this one extra step, as we'll see here. So what we're going to do is we're going to imagine generating these particles one time step at a time. So first, time's on the x-axis, particles on the y. First time step, we've proposed a bunch of particles by sampling from our proposal distribution for time step 1. We're going to weigh them based on the probability at that time step of the data for those particles. and again, normalized by the proposal probability. And these weights we're going to indicate here by the colors. So some of these particles are going to be good. They're going to be close. They're going to assign high probability to the data at this time step. Others not going to be so good. And we're going to try to focus our effort on promising trajectories. So rather than just propagating all of these forward, what we're going to do is we're going to try to focus on the good ones. So, what are these weights telling us? The first part of this is like, how likely was this particle to have continued from from the trajectory up until time t? The likelihood is how consistent is it, or how likely is the data under this particle? Then we have to divide by q in order to normalize, just like we did with importance sampling. Once we've done this, what we're going to do is we're going to sample what we call ancestor indices. So effectively, this green particle here was good. So we're going to want to sample that multiple times. Red particle, not very good. So we're going to want to not sample that with very high probability. That trajectory has reached a dead end. It is not worth our effort to continue propagating that trajectory forward because it's not consistent with either the dynamics or the observations. And so in this way, this is effectively, so what's happening, we sample based on the normalized weights. You can think of this as a distribution over particles. And this is going to tend to replicate good particles and kill off bad particles. So now we can repeat the process. And now these two particles are both following from this trajectory that was good up until time till time t. We do the same thing. We weigh them, compute the normalized weights and sample from this categorical distribution. And here we just propagate the good particles yet again. Do that, rinse and repeat until the end. And what you get is, again, an estimator for posterior distributions, not just over a single variable, but over a whole trajectory. And so, what's going on here is that the posterior, we're saying, is approximately equal to this weighted sum of trajectories. Each x1 through t superscript I is a trajectory through latent space. And each weight here is a weight on that trajectory at the final time step. So, one important design choice that we need to consider here is that proposal distribution Q. So remember, that proposal distribution, we would like to be, putting particles in places where they're going to be high probability under both the dynamics. They're likely to continue the trajectory, but also high probability of the data that we're observing. And so how do we go about choosing Q? Well, one idea is that you could try to learn Q, and while you're at it, try to learn the parameters of your model too. We'll write this algorithm out pretty generically by sort of iteratively running sequential Monte Carlo and then updating the parameters. And one way of doing that is what's called reweighted wake sleep. The idea is pretty simple. The first step is to run self-normalized importance sampling or sequential Monte Carlo with some fixed proposal P and some current model P and fixed proposal Q, and then update the proposal and the model. How do we update the proposal? Well, what we're going to do is we're going to try to make Q look more like the posterior that was estimated from step 1. How do we define more similar to the posterior? There's many ways to say how similar two distributions are. What we're going to do here is try to minimize the inclusive KL divergence, i.e. KL PQ. And we can do that with simple gradient descent. So KL PQ, we want the gradient with respect to the parameters of Q. This is just, KL by definition is an expected value of log P over Q. So the gradient of KL is going to be, we can pass that gradient inside the expectation. And this we can approximate using the trajectories that we've gotten from step 1. Likewise, to update the model P, we can use our particles to approximate the gradient of the KL, but now with respect to the parameters of P. So run SMC, take a gradient step, minimize the KL, run SMC again, more gradient steps, so on and so forth. Do that and repeat until convergence. This is the reweighted wake sleep algorithm. Yeah.
I have a question for SMC. So you said you progress on trajectories, right? But then you're biasing the distribution by throwing it away. So can you still show that as the number of samples goes to infinity, you still have an unbiased estimate?
Yeah, I actually dropped a slide at the end of SMC that I was thinking about saying, which is that you can show that actually this procedure, even though it drops certain trajectories, it replicates other trajectories. It doesn't induce bias into the estimator. You still get an unbiased estimate of the marginal likelihood from SMC, but it has the same sorts of biases of the particle sample paths like we talked for importance sampling as well. And the other thing that is worth noting about SMC is that in terms of variance, you can show that the variance of the sequential Monte Carlo estimators is only growing linearly in the number of time steps, whereas for importance sampling, it's growing exponentially with the number of time steps. And so, yeah.
Quick question. I missed what are bad weights? Are those the weights that are large or small?
Small. Good weights are big.
Okay.
Bad weights are small. Yeah, because bad weights are essentially like particles that have low probability under the dynamics model or under the likelihood.
Thanks.
Are there other questions on SMC or reweighted the wake sleep or anything at this point? Yeah.
The weak sleep part. So on that slide, the steps 2 and 3 are what we call like the sleep part, like after you've done all your sampling part, who are improving Q and P.
Yes. Now, that's my understanding. I honestly don't follow all of the analogy for why they call it wake sleep. I think that it's by analogy to like when you're awake, you're drawing inferences about the world. These are your posteriors over particles, or your posterior particles. And then when you sleep, maybe you're consolidating these into better models of the world, doing some sort of learning during sleep. I think there's a vaguely neurobiological argument for why that might make sense. Maybe that's how the brain works, but I'm not going to make any promises about how the brain works today. Just saying, I think that's why they call it reweighted wake sleep.
I mean, this sounds like actor critical reinforcement learning.
Yeah, I mean, there's a lot of algorithms that have a flavor like this. Is it like actor critic for the Zoom audience? Yeah. A lot of algorithms have some alternating like coordinate update where they say, all right, I'm going to fix my generative model. I'm going to draw inferences about latent variables, and then I'm going to update parameters. That's kind of expectation maximization as well. Here, yeah, there's this extra step of not only do I have to update my model of the world P, I also am updating this sort of proposal distribution, which isn't really a model of the world. It's just this auxiliary thing that I use to perform inference. This proposal distribution that I employ during SMC. Other questions?
Can I ask a question? I'm from online.
Yeah, sure.
The first question is why you can use the importance samples to estimate the expectation over P, because Q is not the same as P. Yes. So
I think the answer there is similar to like an expectation maximization, where what we're doing is we're taking what we did in this bottom step, and I'm sorry you can't see my cursor. Let me use my mouse. In this bottom step, you might be concerned that we want to take a gradient with respect to theta of this expression, but doesn't theta also appear here, right?
Yeah.
So what we're doing is we're effectively freezing this posterior distribution and saying, we're going to split this. First, we're going to estimate this using SMC and then freeze this. These are like the posterior based on my previous parameters. And I'm only going to take the gradient inside this expectation. This is effectively what expectation maximization does too. You could, the notation makes it a little bit harder to see, but you could imagine sort of giving this a different name. I've already used Q. I don't want to use R. S. You could imagine giving this a different name and then say, I'm going to do coordinate descent on S, my posterior approximation, theta, my parameters of this model. And then also, here we've got this extra Q up here for the proposal. So yeah, I see what you're saying, and I can see how it could be confusing. I'm just saying this is a splitting technique that we use all the time in not just reweighted wake sleep, but also in EM.
Okay. Another question is regarding the importance sampling techniques, that page. They claim the optimal Q choice is the posterior P, but I think that's not correct, because you want to can you go back to the previous page? No, previous.
Sorry, which one's previous? Forward?
Oh, yeah,
Maybe you could ask the question, then we
yeah,
can find the slide.
yeah. Like the importance sampling, you're going to estimate the expectation. The expectation is fx times pfx, and you want to use a proposed distribution q. And
Yeah, that's a good point. Yeah, I see what you're
yeah,
saying.
and
Yeah.
I think the optimal of q is actually not the p, but p times f.
Yeah, so you're right. So I was being a little bit fast and loose here. I think this is the slide you want. Right.
Yeah.
Yeah, so if you knew that you wanted to compute the expectation of a specific function f, then you're absolutely right. You would want to propose from something that has high probability under p and also has large amplitude under f. But if you don't fix f a priori, if you're saying I want expectations under sort of any function or any function in some function class, then I think it's pretty safe to say that you would like your proposals to look like your posterior. But you're absolutely right in general for fixed f.
Okay, good. Thank you.
And you're on the right track also, because it's going to turn out that this proposal distribution is really one of the main things that we're going to be trying to learn here, and the object of interest for 6.0. So why I want to come back to reweighted wake sleep here. Oh yeah, sorry, one question.
I'm not sure if it is similar to the question that was asked because I missed the question. So when you take the gradient there, I see that it's gradient with respect to phi, so the expectation doesn't do anything. But here, when you take gradient with respect to theta, you have to take the expected gradient of the probability also.
Yeah,
right?
yeah, that was exactly the question.
that's a-
Two questions
sorry.
before. I was just saying you can think of this as like a splitting technique that we use in EM as well, where we're fixing this distribution and optimizing the parameters, and then fixing the parameters and re-estimating this posterior. So yeah, I will change this slide after because I can see how it's confusing. Yeah.
So you're fitting the proposal distribution, or I guess you're doing gradient descent to try to make it as close as possible to the posterior. But I was just wondering, is it sensitive to your initialization of the proposal distribution? Like, are there certain, do you have to get close enough to the posterior to make this learning process possible?
Yeah, so the question is, how sensitive is this sort of learning of the proposal to the initialization? And I think your intuition is right that, this is going to be a non-convex optimization where, there's a bit of a chicken and the egg problem here. Like, we want to have a decent enough proposal that the posterior overstates the particles that we get out are not like totally often no man's land, or super high variance estimates of this gradient is another way to look at that. So, how do we sort of solve that? And initialization is certainly going to be a part of this. A common thing to do is to initialize Q to what's called the bootstrap proposal, which is really just propose using the dynamics model that you currently have, and then we can hopefully learn better proposals as we go. But one question that nobody asked that I thought might come up is why are we using this so inclusive KL divergence? Why KLPQ instead of KLQP? And one reason for that is that KLQP versus KLPQ have sort of different properties in how they approximate or how they measure distances. So when you minimize the reverse KL from Q to P, the optimal Q is one that puts all of its probability on a mode. It's mode seeking behavior. It wants to find a mode of P. And the reason for that is that P appears in the denominator here. And so if Q puts probability in places where P is 0, you end up with this diverging KL divergence. You get a log of infinity. If you do the opposite, if you minimize the forward or inclusive KL from P to Q, what you get is a log P over Q, and this has the opposite behavior. This is mean seeking and mode covering. And so this gives us generally broader or heavier tailed approximations Q. And that's kind of what you want when you're doing importance sampling. You want to be sort of, you would rather sort of err on the side of being conservative and proposing too broadly than proposing just in one mode and perhaps missing important modes of your posterior distribution. So this is one argument for why inclusive KL. And, if we go back to reweighted wake sleep, I was showing it for the sort of importance sampling case, but if you're in the time series case, the gradients that we need are really expectations with respect to these smoothing distributions over particles. Here I've got Z and X instead of X and Y, but Z up till time T given the observations up until time big T. So, when you put this into the time series setting, SMC kind of gives you what you need to compute these expectations. Now, are we done? Why am I still talking about reweighted wake sleep? What can we go home now? The answer is no. It turns out that for long sequences, resampling was really crucial to getting low variance estimates, but it also induces particle degeneracy at early time steps. So the early time step posteriors, there's only a few particles. The yellow particles are the only ones that survive until time t. And you can see that they all are stemming from a few particles that started out at time 1. So maybe your expectations over the state at time 1 are going to be considerably higher variance because they only have a few particles giving rise to them. So to see a particular case where that failure arises, imagine a very simple model, a chain graph, where you've got latent states following a linear Gaussian system over time, and then you only get an observation at the very end. So in typical sequential Monte Carlo, and the way that I described it so far, what you're weighting those particles by are what are called the filtering distribution. So you have a sequence of distributions, P of x1 and y1 up to P of x1 through n and y1 through n. And in this case, since you only observe an observation at time n, all of those filtering distributions are really just the prior, which is very broad. So if we knew that this linear Gaussian chain started at 0, and then we knew it at time 10, we got an observation at y equals 10, the filtering distributions would look like the prior up until time step 9. These are just Gaussians centered on 0 that are getting broader and broader and broader. And then once we finally get this observation, we see that this filtering distribution shifts way over to the right. And this big jump in distributions from time step 9 to time step 10 means that by time step 9, most of our particles are going to look like this broad Gaussian here, and we're going to get very few particles that happen to be close to this target distribution that we really needed to end up at. And so this is basically doomed to fail in this simple setting. And instead, what we'd like to do is we'd like to sample our particles at each time step from the smoothing distribution that has the advantage of looking ahead and saying, I know I need to end up at time step 10 at the end. So let me make sure that I get some particles, even at time step 1 and 2, they're going to be biased in that direction. If you could do this, you'd be in business. But if you do the filtering distributions like is standard in SMC, you're going to suffer from this particle degeneracy, and especially in this very simple case. So, the trajectories I showed you before were exactly the trajectories from SMC with these filtering distributions. If you use this smoothing method called 6O, you'll find that the particles you get at the end are really expressing or capturing uncertainty throughout the whole trajectory, as opposed to just at the very late time steps here. So, let me just unpack that a little bit more and show you how this works. So the smoothing distributions, these are the distributions over x1 and y1 through t. So the data at time 1 and all the observations up to and including the future observations. Now, if we could target our particles to these smoothing distributions, that would be great, but unfortunately, they're not available to us. In fact, if we had these distributions, then we wouldn't have done SMC in the first place because we'd basically be done. We would just sample from these directly. So these are the things that we would like to target our SMC sweeps toward, but we don't have them. So the idea is, maybe we can estimate this thing in a reasonable way. So can we decompose the smoothing distributions? We can decompose these smoothing distributions into a product of two terms. One is the filtering distribution, so the observations up until time little t only. And then the other factor is this look-ahead distribution, the thing that we need to estimate. So what we're going to do, and what 6O is based on, is just approximating this look ahead distribution with some function R that's going to have some parameters that we'll learn as well. So the key insight into how we're going to learn that thing is we're going to say this look ahead distribution, P of future data given state at time t, we can approximate this thing using density ratio estimation. So first of all, we'll use Bayes' rule. We'll write this as the joint divided by the marginal. Nothing fancy. We'll expand out the joint distribution into the smoothing distribution, or like this, P of X given future data times P of future data. And we'll observe that we actually don't care about this marginal probability of the future data, because we're only ever using this when we're weighting particles, and this is going to be the same regardless of which location X at time t our particle is occupying. So this is just going to be a constant for all our purposes. And so really, we care about this ratio, P of X given future data to P of X marginally. And importantly, we can sample both of these distributions using SMC, or using our prior, we can generate samples of the marginal. Using SMC, we can get samples of X given future data. And so all we're going to do is we're going to train our classifier to distinguish between samples drawn from these two distributions. And it turns out that when you train a classifier to do this, the optimal classifier is one that's going to produce logits that are just the log, it's the log of this density ratio. And this is a commonly used trick for density ratio estimating. Density ratio estimation is turning it into a classification problem. This is exactly the trick that lies behind generative adversarial networks, GANs, things like that. So here we're just using this for this particular purpose of learning a good look ahead distribution. So how does that work? Well, first, the 6.0 algorithm has a couple of steps. We'll draw samples from our prior probability distribution over states. We'll draw joint samples from X and Y using the joint distribution again, and we'll train a classifier with two types of samples, X1 and Y1, which are drawn jointly from this model, and then X tilde and Y, which are drawn independently. This one marginally, and this one, you can think of this as also marginally from P of Y. Then we'll train a neural network, recurrent neural network, to go backward over the samples and produce some hidden state, which we can then plug in to get our estimate of this classifier at each time step. And then finally, once we've done that, we can use it inside of SMC. You could do this inside of a learning loop, just like reweighted wake sleep. There's other methods like what we talk about in the 6.0 paper for doing this. But really, the key idea here is that we've learned this look ahead approximation that's going to allow us to reweight our particles in a way that's going to be consistent with future data, not just with the data up until time t. I'm behind schedule, so I don't want to dwell too long on some of this stuff because it's kind of saying things that I've said already. SMC doesn't suffer from this particle degeneracy problem when you are reweighting in this inside the loop based on future data. It turns out, the previous methods, for example, neural adaptive SMC, use these filtering distributions when learning the proposals, but it biases them in a way. And my student told me I needed to include this meme that he created. Apparently, Gus Fring has a lot to say about smoothing and filtering distributions. So, what's the difference between neural adaptive SMC and the method that we're working on? Well, neural adaptive SMC approximates posteriors with filtering distributions. We approximate them with smoothing distributions. Those are not the same. So if you're on Twitter, you may know what this means. I have no idea, but I do like Gus Fring. And I think that the sentiment is true. This is the main difference, and they are not the same. So, does this all work? We have been testing this out. Now we're kind of moving from 6.0 to some of our very recent extensions of this, the reweighted wake sleep using these ideas. And linear Gaussian systems is a nice playground to check if algorithms are working and how they differ from one another. So what we find is that when you use the new method versus the previous methods, that there's a big difference in the posterior distributions you get and the proposals that you learn, especially in the variances, which is maybe unsurprising. But this is just linear Gaussian stuff. We want to really know in more complex models whether this works. And so, we've been trying this out in switching linear dynamical systems like I showed you yesterday, where we've got both continuous and discrete states. And one of the nice things about this approach is that it works out of the box with discrete latent variable models too, which isn't true of some of the other previous methods. And we find that learning these proposals based on the smoothing distribution makes a big difference, especially in very noisy settings. So when everything is pretty low noise, filtering and smoothing distributions don't really differ all that much because they're dominated by the instantaneous likelihood. But when you've got substantial noise, then incorporating future information really makes a big difference. And so that's kind of the takeaway from this. Finally, let's come back to that Hodgkin-Huxley example. So this is a nice one where we have relatively slow and noisy voltage measurements. From these, we're kind of simulating data here, but we're imagining this is a stand-in for like a voltage imaging setup. When you run the existing methods that just use the filtering distributions, you get a lot of these spurious spikes. They're not particularly well aligned with the true spikes, which are shown in red. And a lot of this is boiling down to just having, a bad collection of particles at some time step because you're not looking ahead into the future to know that you need to have a particle primed and ready to spike in a millisecond or two. And when we contrast that with what we can do with these smoothing-based methods, we see that they're much better at capturing this underlying voltage trajectory. Again, this is simulated data, but this is where we're headed, and we can really see a big quantitative difference in terms of log likelihood bounds that we get using this new method in purple versus preceding methods. One way to read these is that all the methods, if you give them 256 particles, anything works at that point, and that's kind of what you expect. With enough particles, SMC will eventually work. If you only have 4 particles, then it really helps to make sure that you're proposing intelligently in ways that are going to put those particles in position to explain future data. And what we see is that with 4 particles, using the new method, we can do as well as say, 6.0, which is the one that we presented at NeurIPS using 10-ish or so particles, and as well as NASMC using, 128 particles. So there's a real savings in terms of number of particles, and that is going to pay dividends when it comes to trying to learn these models, because every time you want to remember this, like SMC sweep is happening inside the loop of reweighted wake sleep. So every time you want to take a gradient step, you got to run SMC. Running SMC with 4 particles is a lot faster than running SMC with 128 or 256 particles. Yeah.
Some of these other methods that use filtering distributions, can they be run online? Like, is that an advantage that using smoothing distributions doesn't give you?
Yeah. So to do the smoothing, compute the smoothing distributions, you're conditioning on future data to update your particle weights. If you don't have that, then these smoothing-based methods just basically reduce to the filtering-based methods. So I don't think you lose anything, but you strictly gain things when you're doing this sort of offline model learning, where you're running SMC offline in order to get gradients for your parameters and such.
Okay, and my other question was, maybe you're going to get to this, but how do these sampling methods in general compare to like variational RNN sorts of methods?
That's a great question. So we don't have them listed here. We did do comparisons like that for 5.0 and 6.0 papers. And in general, these variational inference-based methods like amortized VI methods that train a RNN to take in a sequence and produce a posterior are going to give you strictly biased estimates because they're not going to have truly captured the true posterior, even in the asymptotic limit, which, here we can always give more particles and say, eventually this thing's going to get closer and closer. And that pays off in terms of these log likelihood bounds. It also pays off in terms of how quickly and how accurately you can learn model parameters. So these SMC-based approaches, I think, are, strictly better than the VI, like with a recurrent neural network encoder-based approaches. I'm not going to get to that today, but that's my take on this. And yeah, where are we going next? Well, we're simulating multi-compartment models, and we're trying to do exactly this type of thing on much more complex settings. Hodgkin-Huxley, one compartment, it's only like 4 state variables, but these methods work on much higher dimensional state spaces. And those are also the places where having good particle proposals really makes a big difference. So yeah, I'm very excited about this stuff. You can read about 6.0 at NeurIPS last year, the neural adaptive SMC with twists, NASTY for short. That's hopefully going to be submitted to NeurIPS this year, so stay tuned. We'll hopefully have that on archive in the not too distant future. Can I take any more questions on 6.0? I think I'm not going to get through all of the S5 stuff before 2:30, so maybe it's best to take a couple questions here and just give you the flavor of S5. Yeah.
I sort of missed what's the difference between NASTY and 6.0? What's the
I didn't really explain that. So 6.0 is basically like SMC with these smoothing distributions, but you can also, for the parameter learning part of that, you can also try to take gradients through the lower bound estimate that you get from SMC using that smoothing or using the look ahead distribution approximation. NASTY is just saying, well, rather than doing gradient ascent on the lower bound from 6.0, let's do this reweighted wake sleep thing. And it turns out that is a really simple idea that we should have had a long time ago, because it works considerably better than just doing the gradient descent based approach. Good. All right, let me, in the last 10 minutes, just give you a bit about S5, because it's actually a very simple idea. And even if we don't get all the details, then at least you'll get the gist. This is work from students Jimmy and postdoc Andy, and the motivation is from this, from a canonical problem in neuroscience that Chetan's lab has really done a great job to make benchmarks around. So they have what's called the neural latence benchmark, and one of the key tasks in that is to be able to take in a spike train from some subset of neurons and output predictions about firing rates for held out neurons. Now, ideally, we'd like to do this in a way that is based on some model that's built out of pieces that maybe we can sort of reverse engineer and understand how they're making these predictions. And so a lot of work in neuroscience goes into building models that can address this prediction problem, but in a way that's sort of interpretable. State space models are canonical ways of doing this. You could do this with a linear dynamical system, where first you try to infer some low dimensional latent states from the held in neurons, and then based on those latent states, make predictions about firing rates for held out neurons. This is a common approach. This is a standard baseline that's included in this benchmark. And over the past couple of years, there's been a lot of work to try to advance and improve upon these baselines with more flexible models, and then hopefully models that, like I said, we can go in and we can try to understand post hoc. The way that S5 works is built on pieces that we know and love. It's all built around simple linear dynamical systems. Here we have a continuous time linear dynamical system. So state X, continuous time dynamics, X dot equals AX plus BU. U are some inputs to the system, and Y are the readouts or the emissions. They're a linear function of X, maybe also a linear function of the inputs as well. So this is a building block for so many of the models that we use on a day-to-day basis, many of what I showed you yesterday. We can discretize that continuous time system in a number of ways to get a discrete time linear dynamical system, where now we'll use K to index time. So here, we're just using what's called the zero order hold discretization. That's not particularly important. This is just saying this one way to turn this into this graphical model like we've seen a number of times. One thing to note, we're not going to have any stochasticity in these dynamics or in these emissions. That's an interesting direction for future work, but so far, and in this, what I'm talking about now, they'll all be deterministic. Now, linear systems are great. I've said this, I think I said the exact same words yesterday. Linear systems are great, but there's only so much you can do with them. One thing that you can do is you can extend them with nonlinear dynamics, like we talked about yesterday. Another thing that you can do is you can extend them with nonlinear emissions. And a simple way to do that is to pass the outputs at each time step through some nonlinearity f. And this f could be sort of arbitrarily complex, as long as it, in this setting, only applies to a single time step at a time. In our setting, we'll just use very simple rectified linear GLU activations, but you could get much fancier here. Once you've got a layer like that, the key idea behind the predecessors to S5, namely S4, which is work by Albert Gu, is to just stack this on top of itself. So if you want to build a thing that takes in a sequence and produces a new sequence, well, this model does that. It takes in a sequence of inputs and produces a sequence of outputs, but it still is fairly limited in its expressivity. It's just a linear dynamical system with nonlinear transformation of the outputs. You can get much more flexible models if you stack these on top of each other. You just have a stack of linear state space layers with nonlinearities in between at each one. There's some reasons why you might expect this to be able to capture nonlinear dynamics, even though it's only got linear dynamics at each time step. Again, we're not going to get into all the details on that, but this basic building block is one that's sort of incredibly simple, and as we'll see, surprisingly, it actually works really well for a number of problems. One thing to note, and it's one thing that's nice about this formulation, is that when you have a state space layer, you can equivalently represent it as a convolution. And this is nothing deep. This is, signal processing 101 stuff. What I'm saying is that you can write the output y as a linear function of the inputs up until time t. So y1 is some function of u0 and u1. And the way you get that is just by expanding out the definitions of these deterministic recursions, as we're seeing here. And once you've written it that way, you can represent the output at time step k as a linear function of all of the inputs up until time step k. And you can represent that as a convolution. And this convolution kernel here is just this, this repeating pattern of emission matrices times dynamics and input matrices. And this is nice because there's all sorts of fast convolutions built into deep learning libraries. So if we can turn this from a recursive linear system into a convolution, then we can kind of take advantage of those built-in primitives. The problem is that in order to even construct this convolution kernel, you got to do a lot of work. You have to multiply these matrices to these powers, and you got to do different powers for each time step in this kernel. Albert Gu, who did the preceding work on this stuff, did a lot of interesting mathematical work to show how certain types of dynamics matrices allow you to compute this thing very efficiently, but it only works for one-dimensional inputs and one-dimensional outputs. So there were some pretty strong constraints, and even still, in order to show that this works, you had to appeal to stuff about Cauchy kernels and do everything in frequency domain, stuff that is certainly doable. It's not like no individual step is particularly hard, but there's a lot of simple, slightly challenging steps put together adds up to a rather complicated process. And in particular, the model at the end involves running convolutions across each dimension of input in parallel in order to implement one of these layers. And the computational steps within each layer involve a number of steps to convert things efficiently into Fourier domain, run these convolutions as multiplications, and invert them back out. The net result is something that's very efficient. It can be done in ways that are linear in time or linear or logarithmic in time if you have a parallel machine. So it's very fast in the end, but it's rather complicated. Advantages and disadvantages, again, in the interest of time, I won't dwell too much here. I'll just say, my take on this, when I read about this, I was like, man, that is such a cool starting point. I love the idea of just stacking these linear systems with a little bit of nonlinear glue in between. But this implementation is a lot to digest. If you read the paper, there are many moving pieces. So we thought, can we do this in a way that's simpler? And that's what led us to S5. And the idea was, let's get rid of all this, one-dimensional convolutions and per input channel filters. let's remove some of the constraints that were inherent in that, and let's do everything in the time domain, and let's not sacrifice anything on performance or efficiency. And that's what we call S5. The model itself gets rid of these, per input channel dynamics and just has like your standard linear dynamics, just like before. And in terms of the computation, instead of all this stuff with Cauchy kernels and whatnot, all we do is we diagonalize this system and we run what's called a parallel scan. Now, if I had a bit more time, I would really walk you through all the details of this. Parallel scans are, again, textbook things that computer scientists have known about, numerical algebra people have known about for a long time. All they do is they take this linear recursion that looks like it should take order L time, and on a parallel machine, they are able to perform this in only logarithmic time. So to give you one example of that, like suppose that you want to just sum up the numbers A, B, C, D. You could do that linearly, AB plus C, AB plus C plus D, so on and so forth. That would take you 3 steps, but you could do it in parallel too. And to do it in parallel, you would add both of these, and then you would add up the results of those. And as long as you have sort of order L processors, you can do this in order log L time, which is really cool. Turns out you can do the same thing for a linear recurrence. So if you want to compute these outputs for 4 time steps, you could do that linearly, or you could define a binary associative operator that takes in a tuple like this, and then outputs a tuple of the same type. There's a little bit of math you can do to convince yourself that at the end, this is producing all the x's that you want as one of the elements of these tuples, and it does so in order logarithmic time. The main bottleneck, though, is that you end up with these matrix multiplications, and this is where that diagonalization step really comes in. You don't want to multiply dense matrices together a bunch of times, but if they're diagonal, who cares? It's no big deal. So you can implement all this in only 70 lines of code, and most of these lines are comments, and I think that's a pretty good sign that this is a simplification. You don't have to write any custom CUDA kernels, for example, which is not a thing anybody should do anymore. So again, I want to wrap up in just 2 minutes here. It works really well. It works on long sequence benchmarks, which are a little bit artificial. They often involve like raveling an image. So take this image, which is maybe, 32 by 32 pixels, or 100 or 64 by 64, something like that. And then the question is, are these 2 dots connected by a path? And here the answer is yes. Here the answer is no, they're not connected. It's actually is kind of a challenging problem for me to solve looking at the image. And you can imagine trying to solve this if you just got a sequence of pixel values that is like 16,000 time steps long. So on this benchmark, transformers, state of the art in many sequence to sequence modeling tasks for large language modeling, won't even run because the context length is too large. But these state space layers are essentially nailing the problem, which is really pretty interesting. And for a long time, or for a little bit of time, S5 was actually leading on these benchmarks, and then Meta, Facebook came out with methods that, using a little bit more memory, eke out a small amount of improvement. But we're still, doing very competitively, especially on this hardest PathX sequence to sequence task, which I think is pretty cool. And we've done it in places where you have irregularly spaced samples, which is one of these things that you couldn't really do with S4 because you had to construct this convolution kernel that applied at all time steps, and that doesn't work if your time steps are irregularly spaced. But it works fine when you do everything in the time domain, and it's like 130 times faster than the preceding methods for this, and also a lot better. And on the neural latence benchmark that had motivated this, how does this compare conceptually? Well, it's got linear dynamics in time, nonlinear in depth. Contrast that with RNNs or transformers, which have somewhat different architectures and assumptions. And on these benchmarks, S5 is also doing really well. And so we're pretty excited about this, and we're just starting to dig into how it's doing those making those predictions and where it's getting that improvement from. But the initial results are promising that, yeah, this is not only a method that's accurate, but also one that's, it's built out of linear systems. Linear systems are things that, we have a lot of experience with, a lot of intuition about. Can we use that to better understand how these predictions are made? So I'm excited about this. Again, if you missed yesterday, check out the code that we've been writing for working with state space models. And I want to just say thanks again to you guys for inviting me for these sequential lectures. It's been really fun. And thanks to my group for all the hard work that I was able to talk about today. Thank you.
We're almost out of time. Maybe we'll take just one quick question. Okay, if not, let's thank Scott again.
Yeah.